Okay, so this is a revision video on monoclonal antibodies and their use in diagnosis. We're focusing in this video on ELISA. So the ELISA is the enzyme-linked immunosorbent assay. So there are 3 revision tasks. Pause the video, complete those tasks, and then we'll run through the answers. Okay, so question 1, how are monoclonal antibodies used in an ELISA? And we're specifically looking at diagnosis of HIV because that's the example you're expected to know about. So let's just talk through how this happens. So first of all, a sample has to be provided. So in the case of HIV, that will be a blood sample. And that sample will be centrifuged. And so red blood cells, those heavier parts of the blood, will move to the bottom of the tube, and at the top you will have the plasma. And that's where the sample's taken from, the plasma. And within that plasma there will be the antigen. So in this example it's obviously HIV. So a sample will be taken, and then what's commonly done is that many tests are run in parallel. So you'll have this plate that has lots of wells on it, normally 96, and then you can add 96 samples, and you'll obviously want to repeat some of those samples to check that your result is correct. So sample will be added to one of those wells. So let's focus in what happens in one of those wells during the ELISA. So in this example we've got the well here, the plasma sample has been added and it's got HIV in the sample. So the first thing that happens is this. It's applied to the surface and it sticks to the surface. And then what happens is this. So any unattached antigens are washed away. Obviously there's going to be many other proteins in the sample that are going to attach, but we're just interested in the HIV. The next step is this. This is the first use of the monoclonal antibody in this ELISA. So one that is specific to the antigen. So in this case, HIV. This is sometimes called the primary antibody, and that will bind. The next step. So any excess antibodies washed away, only the antibody that's bound to the antigen is left. And then the next step comes along, which is this. So at this stage, another antibody is added. That's called the secondary antibody. And the key thing about the secondary antibody is that it has an enzyme attached to its heavy chain. And it binds to that primary antibody. So now, what happens next? So a colourless substrate is added for that enzyme. And then if there's enzyme present, that will obviously catalyse the conversion of that substrate into a coloured product. So the intensity of that colour can be used to measure the amount of antigen present. So that's basically how an ELISA works and how monoclonal antibodies are used in an ELISA. This is one example of a type of ELISA. There are other ways an ELISA can be conducted, but it's still using ultimately the enzyme-linked secondary antibody. So this is a useful example. This is the example that's actually given in the textbook, but beware that in exams you may be asked how an ELISA works in a slightly different way, but the principle is the same. So let's move on to answering question 2 and 3. So why could the HIV test not be used to diagnose another disease? And then to name 2 other diseases that can be diagnosed using ELISA. Here are your answers. So obviously for question 2, because the antibodies in the ELISA are specific only for HIV, you would only be able to diagnose HIV. Any other antigens present, those antibodies will not bind to. So you couldn't use it to diagnose any other disease other than HIV. And question 3, many diseases, but some examples are flu, hepatitis, and cancer. And a common example is prostate cancer, where the levels of PSA become very high in the blood. Okay, so that's the ELISA test.